Okay, students. So let us continue the NEET and CET questions of the chapters Sexual Reproduction in Flowering Plants. In last class we discussed around 36 questions. Now let us continue the questions. The next question, 37th question. There are two statements. We have to go through these statements and we have to judge which is whether both the statements are right or both the statements are wrong, or we have to judge which statement is right and which statement is wrong. Okay. According to statement 1, sporopollenin can withstand high temperature but not strong acids. So sporopollenin is a hardest material present on the exine wall of the pollen grain. So as well aware, these pollen grain, because of this presence of sporopollenin, they can be preserved as fossils. So sporopollenin is a very hard material present on the wall of exine, and it is resistant to high temperature and even strong acids. So statement 1 is not the right statement. It can resistant to strong acids also. The second statement, sporopollenin can be degraded by enzymes. No, sporopollenin cannot be degraded by the enzyme. Many of the animals consume the plant inflorescence, etc. Even in their dung, we can recover the pollen grains which are viable. So, for this question, both the statement A and both the statement B is wrong. In this type of question, we should be clear. We should not find the option. By reading the statements itself, we should understand whether the statement is right or not. So, option is both statement A and B are wrong. Next one. Next question. In angiosperms, functional megaspore develops into. So we have discussed earlier. So this is the ovule. So in the nucellus, a single cell present towards micropyle develops into megaspore mother cell, and this megaspore mother cell, on meiotic division, it produces 4 megaspores. Out of 4 megaspores which are haploid, megaspore mother cell is diploid. On meiotic division, it produces 4 megaspore which are haploid. 3 will get degenerated. Only 1 will become the functional megaspore, and this functional megaspore give rise to the female gametophyte, what we call it as embryo sac. Thus, we stated that because the embryo sac or female gametophyte is developed from single functional megaspore, this development is also called monosporic development. Let us go through the question once again. In angiosperm, functional megaspore develops into embryo sac. It's not the endosperm. It is embryo sac. In the embryo sac, after fertilization, the 2 polar nuclei fuses with the male gamete to form primary endosperm nuclei, and then which develops into endosperm. So the answer is embryo sac. Next one. So arrangement of the nuclei in normal dicot embryo sac is seen. Arrangement of the nuclei in normal dicot embryo sac. So embryo sac is 7-celled 8-nucleate structure. So towards micropylar end there are 2 synergids and 1 together, call it as egg apparatus. So in the center there are 2 polar nuclei inside the central cell, and towards the chelazal end there are 3 cells which are called antipodals. So 3 plus 2 plus 1. So the option is 3 plus 2 plus 3. Sorry, not 1. It is 3 plus 2 plus 3. 3 are the egg apparatus, 2 synergid and 1 egg cell. 2 are the 2 polar nuclei present in the central cell. And 3 are antipodals. So the option is the C. Next question. So here they are given a figure of a plant. They have marked A and B. So this we often leave in the textbooks, considering it's not the figure of show value. But these diagrams are very important. Just you will know how it is important. Generally, we don't concentrate on such type of figure, but many question features in these diagrams also or figures also. Recognize the figure given below and select the correct option. They are given the figure of a plant. They have labeled A and B. So we have to recognize the figure. So recognize the figure and correct option. A, they have claimed that is a chesmogamous flower and B is a clistogamous flower. In option B, A is a clistogamous flower, B is chesmogamous flower. Option 3, A is clistogamous flower and again they have given B is also clistogamous flower. And in option 4, A is chesmogamous flower and B is chesmogamous flower. No. See, in plants like Viola camelina and oxalis, the single plant produce 2 types of flowers. One is the chesmogamous flower, which have exposed stamens and stigma like flowers of other plants. The other one is the clistogamous flower, so which never open until the pollination is achieved. So here A is the chesmogamous flower and B is the clistogamous flower. So the wonderful example to quote, you might have seen the groundnut plant, Arachis hypogea. If you observe the developing stage of that plant, you can see flowers appear, but gradually after some days you may not see the flower, but these flowers penetrate into the soil and after they are the clistogamous flower which are found towards base of the stem and that itself develop into groundnut. So A is a chesmogamous flower and B is a clistogamous flower. Next. Next question. Development of seedless fruits in an unfertilized flower is called. This you already thorough in first year itself. Development of seedless fruit. So these seedless fruits are called parthenocarpic fruit. So unfertilized flower is called. What is parthenocarpy? So the development of the fruits without fertilization is called parthenocarpy and such fruits do not contain seeds. And natural parthenocarpic fruit, you already learned, it is banana. And in horticulture, they induce artificial parthenocarpy by using plant growth regulators or phytohormones. So development of seedless fruit in an unfertilized flower is called parthenocarpy. So parthenogenesis, we know parthenogenesis is development of the female gamete into an offspring without the fertilization or without the contact of the male gamete that takes place in animals. Polyembryony, we already understand presence of more than one embryo inside the embryo sac is called polyembryony. Apomixis, the development of embryos without fertilization is called apomixis. Okay. Germ pore present on the surface of the pollen grain represent. So once again, let us go with the figure diagram. See, pollen grains are covered by 2 wall layers. The outer one is called exile and the inner one is called intile. As exile is deposited by hard, highly resistant material called sporopollen, and at certain region there is no deposition of sporopollen. So you can directly see the intine wall, and these are called germ pores. As soon as the pollen grain lodges on the stigma, if it is a right and compatible pollen, if it gets lodged in right viability period, so the chemicals present in the pollen grain and the stigmatic surface interact with each other, and the pollen tube start developing, and this pollen tube penetrates the stigma style and reaches the ovary. So this pollen tube, generally the entire wall at the region of germ pore bulges out to form pollen tube. So here the question is, germ pores present on the surface of the pollen grain represent area where exine is absent, specialized thickening of exine, specialized thickening of intine, all the above. The answer is option A, area where exine is absent. Next. What does the filiform apparatus do at the entrance into the ovule? It brings about the opening of the pollen tube. It guides the pollen tube from synergate to egg cell. It helps in entry of the pollen tube into synergate. It prevents the entry of more than one pollen tube into embryo sac. So, the main function of the filiform apparatus, which are the differential wall thickening present in the wall of the synergate. See, if you consider there are two synergates, so there are wall striation, these are called filiform apparatus. So the option is, it guides the pollen tube from synergate, it helps the entry of the pollen tube into synergate. Just it helps the entry of the pollen tube into the synergate. So the option is the third option, C. So next there is a mark the following. So this we will neglect. So there are three plants which we have taken as example for plants which producing two types of flower, chesmogamous and clistogamous. So viola, camelina and oxalis. This viola is commonly called as common pansy. So common pansy is viola. Marine grasses are zostera, where we observe hydrophily. That is, that pollination takes place through water. So tallest flower is amorphophilous flower. Likewise, freshwater angiosperm is a Vallisneria, which is also get pollinated through the medium called water. Next slide. So you search the option. Date palm prevents. See, both in date palms and papaya, the male plant bearing male flower is a different one. The female plant bearing female flower is a different one. So in date palm prevents, there is no chance of autogamy because male plant is different, it produces only pollen grains. Female plant is different, it produces ovules which contain female gametophyte called embryo sac. So it prevents autogamy and even geitonogamy. So the option is both A and B. The only chance of pollination in these plants in papaya and date palm is xenogamy. Next slide. Continued self-pollination results in. So in the plants, even in animals, in plants continuous self-pollination, in animals breeding the livestock belonging to same ancestors leads to inbreeding depression. So the answer is direct answer is inbreeding depression. Next question. Pollen grains are generally sticky. See, we have studied in case of wind pollinated plants, the pollen grains are very light in weight and they are not sticky. So they are easily carried by the wind carried without getting stick to any surfaces. In water pollinated plants, pollen grains are very light. They are coated by specialized cell wall material which avoids these pollen grains from wetting. In animal pollinated plants, the pollen grains are sticky because they have to stick to the body of the visiting animal. When this insect or animal visits another flower, these pollen grain may get lodged on the stigma, achieving the pollination. So the option is the C, animal pollinated plants. Next one. Next question. Which statement is not correct? They have given the 4 statements. We have to judge which statement is not the correct one. Pollen grains in water pollinated plants are long and ribbon-like. Of course, yes. This we studied in sea grasses like Zostera. Here both male and female flowers. So in these plants, both the flowers are submerged. The pollen released are very flat and it's ribbon-like, and it swirls in the water current and somehow reaches the stigma. So yes, the first statement is the correct statement. All aquatic plants uses water pollination. No. Only 30 genera of the plants, especially among dicotyledons, they use the water as the media for pollination, not all the aquatic plants. So for that exceptions, we studied Valisteria and Zostera. Apart from that, water hyacinth and in lily, the female flowers emerge out of the surface of water and pollination takes place either by wind or some insects. So not all the aquatic plants use water for pollination. The next, this is the wrong statement. In Valisteria, the female flower reaches the surface of water with long stalk. Of course, yes, the female flower produces long stalk so that the flower reaches the stigmatic surface, reaches the surface of water. When the pollen release, the pollen carried by the water current will reach the stigma and achieves the process pollination. In sea grasses, female flower remains submerged in water, we already did. So the option is, the second one is not true. All the 3 statements are true. Next question. The figure, they have given a figure. So in this figure, representing, we can easily say, but in textbook we don't concentrate about this figure. Generally we don't concentrate. So here you can see number of tassels has come out. So these are nothing but they are the style and stigma of that particular plant. So the option is a wind pollination plant, a water pollination plant, an insect pollination plant, a bird pollination. No, it's a wind pollination plant. So those flowers which are adopted for pollination through the wind, they produce numerous of minute flowers packed into inflorescence. So their ovary generally contains single ovule. They produce exposed stamens and feathery stigma to capture the pollen grain that carried through the wind current. So as we see in comb of this maize plant, so the option is a wind pollinated plant. Next question is, the phenomenon wherein the ovary develop into fruit without fertilization is called, you already know, it's called parthenocarpy. Again, we have to go through the 2 statements and find the correct answer among the 4 options given below. Okay. Enzymes can degrade the organic material of pollen grain exile. No. Pollen grain exile, as we already dealt in the previous question. So it's made up of strong material called sporopollen and it cannot be degraded. Statement B. Sporopollen can withstand high temperature, strong acids and alkali. So both the statements are the right option. So both statement A and B, both statement A and B are correct. This is the second option. Okay. The early stages of embryo development. So what is embryogeny? The early stages of embryo development, that is after the formation of zygote. So the early stages of embryo development is called embryogeny, are dash in both the monocots and dicot. So monocots and dicots differ each other in many characteristics, but when it comes to the embryogeny, it's similar. So the option is the B1, it is similar. Next question. During embryo development, heart-shaped embryo stage is followed by, first, before the development of the zygote into embryo, a small amount of endosperm is formed because endosperm provide nourishment to the developing embryo. So first, after endosperm is formed, by deriving the nutrition from the endosperm, the zygote develops into, first into proembryo, then globular embryo, okay, and after the heart-shaped embryo, it develops into a mature embryo stage. Next question, perisperm. See, this is most important. See, inside the ovule, there is nucellus. This is the nucellus. And embryo sac or female gametophyte is developed towards micropylar end, which is 7 cell 8 nucleate structure. So after fertilization, the egg develop into zygote and polar nuclei with the fusion of one male gamete develop into triploid primary endosperm nuclei. So embryo starts developing, endosperm is formed, embryo develops, and the ovule becomes seed, ovary becomes fruits. See, these seeds may categorized into albuminous seed, which have retained the albumin, and non-albuminous seed. So entire nucellus will be degraded or consumed. But in certain seeds like beet and black pepper, inside the seeds also there are remnants of nucellus, and that remnants of nucellus in the seeds as absorbed in black pepper and beet is called perisperm. Okay, the options are like this. Perisperm is remnants of nucleus, not nucleus. Remnant of nucellus, yes, it's right. Remnant of endosperm, all the above. The option is the B. Next question. Here is the math of following. So it's very simple. They are given only 3 options. You choose the option. Water pollination hydrilla, wind pollination corn, and insect pollination yucca. See, one thing. Here, one important explanation you have to hear about yucca plant. So yucca plant and a particular species of moth and yucca flower, they have the symbiotic relation. So this moth will lay the eggs inside the locule of the ovary because it is the safest place which cannot be predated by predators. So oviposition by the moth takes place inside the locule of the ovary, which is the safest place. By this, the moth is benefited. During the process of laying eggs or oviposition, this yucca flower get pollinated. So this relation is a symbiotic relation. So that you have to remember. So this is the option you search down among the 4 options. Water pollination hydrilla, wind pollination corn, and insect pollination yucca. 56th question. Seed offer several advantages to the angiosperm, which is not true of this. Yes, seeds offer several advantages in angiosperm. Let us read one by one. Since pollination and fertilization are independent of water, seed formation is more independent. So, this is the wrong option. Seed formation is dependent because after fertilization, seed is formed. As the seed, the process of seed formation continues. The seeds remove the water content. It will get dehydrated and it retains only 10 to 15% of the water content. At that time, the embryo present in the seed will go to a period of rest, and that is called dormancy. Again, when we plant a seed, what happens? It absorbs the moisture content through the micropyle. Hydrolysis of the starch takes place. So this supplies the energy and this seed sprout and germinate into new seedling. So water plays an important, it's not an independent phenomenon. Seeds have better, provides protection. So this is seed coats, provides protection to the young embryo. Of course, so that you have to correct it. Seeds are protected by 2 layers of seed coat. Outer testa and inner tegmen, they protect the developing embryo. The odd seed coat provides protection to young embryo. Something they have mismatched the options. Even that is right. Seeds generate new genetic combination leading to variation. It's a product of sexual reproduction. It brings about new genetic recombination. So the option is, which is not true, is, so the first option A. During microsporogenesis, that is the process of production of microspore by the meiotic cell division of microspore mother cell or pollen mother cell, meiosis occurring during microsporogenesis, meiosis occurring. So endothecium, microspore mother cell, microspore tetrad, pollen grains. No, the option is the B. Single microspore mother cell which is diploid undergo meiotic cell division to produce microspore tetrad. On dehydration, they become pollen grains. The option is B. Here again they have given the figure of embryo development. Okay, the question is, which of the following structures shows globular embryo? See here, it is a globular embryo. So this is the early stages of embryogeny, globular embryo, heart-shaped embryo, finally mature embryo. Okay. So next question. In transverse section of typical microsporangium appears dash in outline. This is a very easy question. When we take a TS of anther, there are 4 microsporangium. Each microsporangium is more or less circular in outline. So answer is circular. Next question. Which represent the male gametophyte? So, which is the female gametophyte? Embryo sac, which is 7-celled 8 nucleate structure. Which is the male gametophyte? Pollen grain. Pollen grain is called reduced male gametophyte. It encloses 2 cells. It shaded 2 cells in 60% of the plant. One large cell called vegetative cell or tube cell, and the other one is a small cell which floats in the cytoplasm of vegetative cell called generative cell. So here which represent the male gametophyte. So it's not pollen tetrad, it's not pollen mother cell, it's not sporogeny, it's nothing but pollen grain. Pollen grain is a reduced male gametophyte. Parthenium is also called as carrot grass, which I have already discussed. Next slide. Megasporangium is equivalent to, female gametophyte is the embryo sac, male gametophyte is the pollen grain, ovule is called megasporangium. So answer is the option 3, ovule. Next, in embryo sac, the cell that degenerate after fertilization. See, inside the embryo sac, you are already aware of this. This is the egg apparatus, 2 synergids and 1 egg cell. So this is the central cell containing 2 polar nuclei, and these are the antipodals, 3 antipodals, 7-celled 8 nuclei. See, one sperm fuses with the egg to form diploid zygote. The other male gamete fuses with the 2 haploid polar nuclei to form triploid primary endosperm nuclei. As soon as this double fertilization and triple fusion takes place, both the synergids and antipodals will get degenerated. Here, let us find the option. In an embryo sac, the cell that degenerate after fertilization are synergids and primary endosperm. No. Synergids and antipodals. Yes. Antipodals, primary endosperm. No. Egg and so the option is the B. Synergids and antipodals. Synergids and antipodals. So that is the right option. Next slide. enclosed within the integument is a mass of cell called. So inside the ovular megasporangium, within the integuments, here one point you have to note down. Integument may be one layer or two layer. So integument encloses a mass of nutritive tissue that is called nucellus. Next slide.